Dr. Jain on behalf of PHO chapter. We run this academic program of once a month international speaker program. And Dr. Badira, Dr. Vinod helped me coordinate this program. And because of the midterm CME, we didn't have a session last month, but we are back again and with a very interesting session. I hope you are enjoying this international speaker program, and we are going to bring many more interesting topics for all of you. So over to Badira to take it forward.
Yeah. Thanks, Manas. So, for all those who have joined right now, so this topic today is something different or off track. I mean, you can say to an extent, because this is not something we pediatric oncologists on a routine clinical basis think of, because we all think of the cancer cells and trying to kill those cancer cells with cytotoxic chemotherapy. So this actually steals the limelight from cancer cell to the cells around and the environment around the cancer cells, and trying to attack them. So that's where the whole thing of tumor microenvironment, and we have none other than one of the pioneers who have did so much work in tumor microenvironment, and he has so many cited publications, mentored so many students, and he's one of the oncology luminaries with so many honors and awards. I mean, I can go and on, because if you open up his page, you can see so much there. I would suggest all of you go through that too. So we have Dr. Rakesh Jain with us today, and a person who's very passionate about this. And I think it is a bit contagious too, because even Dr. Bhanavali is as passionate as you about tumor microenvironment and the related things. He's Andrew Work Cook Professor of Tumor Biology. He's Director of EL Steele Laboratories at Massachusetts General Hospital in the Harvard Medical School. And he'll be talking today on, or giving us an overview, and also going into the nuances of tumor microenvironment and how you can reprogram it to and make it better utilized to improve outcomes in cancer. And so this is all about translational medicine from bench to bedside. So I think it's over to you. So, yeah.
Thank you very much for such a kind introduction, and also for your, please convey my thanks to Dr. Banavali for his kind invitation.
Will do.
It's always a pleasure to come back and speak to the physicians and scientists in India. Being an Indian, you can see this is a real pleasure. So what I'm going to do today is talk about, as Badri already said, about tumor microenvironment. So just to give you an idea, many of you are, I know, practicing physicians. So let me just take you to some basic work. So about 200 years ago, a very well-known British pathologist analyzed hundreds of biopsies in London, in UK, and said that he came to a conclusion that that cancer cells are a seed, or assume they are seeds, and then the area around it, the tissue around it, is what he called the soil. And his point was that the seed will not grow until the soil is hospitable. The soil allows it to grow. So as we all know, that most of the cancer field research in the last several decades has focused on the seed, the cancer cell, and did not pay attention to the soil and the seed needs to grow. And what we have learned in the last 20 to 40 years, that the soil, the microenvironment, is as important as the seed. And indeed, this work has been recognized, including by a Nobel Prize in immune checkpoint blockers. And what I'm going to do is talk to you about this because this is something you usually do not hear about or read about or pay attention to. But what I'm going to do is show you that it makes it and how we have taken our findings from bench to bedside, from laboratory to patients. These are my disclosure information. Those are required that I do this for as per Harvard Medical School faculty. So as I was saying to you that in the last several decades, I've seen two revolutions in cancer treatment. One is what is known as genome-driven oncology, where, as Dr. Parmil mentioned, you target cancer cells. You target either chemotherapy or targeted therapy, the mutations in cancer cells. And this therapy in the United States in about 2018 helps only 5% of the patients. And remember, US is the most advanced country in terms of medicine. And only 5% of the patients benefit from targeting the seed. That's really sad, isn't it? Only 5% of the patients benefit from targeting the seed. But if you look at soil, and one of the interesting findings, which I'll talk about, is the immune checkpoint blockers. They benefit in a short period of time in 2018, about 13% of patients. And assume this 2014, 24. So even assume the number has doubled. So it's only 10% of the patients of solid tumors benefit from this approach. But this number is now around 20%. So what I'm trying to say to you, targeting the microenvironment is very critical. And that's where we're going to make a big difference in improving the treatment outcome for cancer patients who have solid tumors that account for a majority of cancer deaths. Even though in the pediatric tumors, the major progress has come from targeting cancer cells in leukemias, the cells that do not have much of a microenvironment, stroma. But not much progress has come in treating solid tumors, as you know, such as medulloblastoma or ependymoma. So these tumors and other, along with the adult solid tumors, microenvironment is a key barrier. And what I'm going to convince you in the next hour is that the TME refers to abbreviation of microenvironment, tumor microenvironment. What I'm going to try to convince you by showing you both preclinical and clinical evidence that tumor microenvironment is abnormal, and this abnormality is what contributes to resistance to every known therapy. So what's tumor microenvironment? So we think about, when somebody says somebody has cancer, we think about primarily about cancer cells, which are shown here in the gray color. Which is great for leukemia, because these are the majority of cells. But in a solid tumor, such as a sarcoma or medulloblastoma, these cells are a very small part of the tumor. A tumor contains, in addition to cancer cells, a variety of host cells of the patient, and they include fibroblasts, various types of immune cells, and all these cells are embedded in extracellular matrix, and they are nourished by blood vessels. That's what brings nutrients and drugs to these cells, and they are drained by lymphatic vessels. So in the last 4 decades, our laboratory has tried to understand the role of each of these compartments, blood vessels, lymphatic vessels, various host cells, and the matrix. And what I'm going to do today is show you how the abnormality of each of these components confers resistance to every known therapy for solid tumors, not so much for liquid cancer. And what I'm going to show you is that we can reprogram it or make it close to normal, then we can improve many treatments. So how do we know about this? So our laboratory at Harvard, at Mass General Hospital, has developed windows in various organs of mice. So this is a brain window where we can visualize a brain tumor growing in a mouse. And these are the blood vessels in that particular tumor. So these are the normal blood vessels, and the tumor is going in this little quadrant right here. And you can see the blood vessels just with naked eye at this magnification look different. Now we have developed microscopes so we can look at a higher magnification, what's going on in these blood vessels. So for example, this is showing the blood flow in these tumor vessels. And you see here the blood flow is quite brisk in these vessels of a tumor. But if you just go slightly here, another region nearby to the tumor, there's no blood supply. So you can imagine you take any chemotherapy, you take any targeted therapy, you take any immune therapy or cell-based therapy, it's going to get here very nicely, no problem. But here it will not. It has going to have to get there by the process of diffusion, which is very slow. Now, but what is interesting, this abnormality is not only in the primary tumor, but also in metastatic sites. So, for example, what we did is we took cancer cells from the same dish, in a cell culture dish, put them in the breast of a mouse, brain of a mouse, and the back skin of a mouse. And then we looked at the blood vessels in these three areas. So you can see they all, but what is one thing which is common in all of these blood vessels? They are highly abnormal. Notice that this tumor has no blood vessels here. And here, blood supply is pretty good, but not here. And this tumor, you see this heterogeneity? So you can imagine why response to solid tumors is very poor because of this inaccessibility of drugs to these regions. But not only are the blood vessels impaired, are defective in tumors, so are the lymphatics. So for example, this is the normal tail of a mouse. And now if you do something known as lymphangiography, where you inject a 2 million molecular weight dextran into the skin, and what you see, that you can see these lymphatic vessels very nicely. They look like chicken wire mesh. But now if you grow a tumor on this tail, a sarcoma, what you see is this is the sarcoma. You see you cannot see any green color because that's what's happened here is that the lymphatics have stopped functioning. And the peritumor lymphatics have become hyperplastic. That means they become bigger. So what happens as a result is the tumors develop very high fluid pressure. So in normal breast or normal skin, the pressure in the tumor, interstitial pressure, is around 0 millimeter mercury. But you look at any type of tumors, brain tumor, sarcoma, lung cancer, you name it, the pressure becomes very high. And whether you look at the primary tumor or metastasis, in all cases. And what this pressure does is pushes drugs out of the tumor. And so what our work of nearly 2 decades has shown, that abnormal blood vessels, the dysfunctional blood vessels, lead to hypoxia, that means low levels of oxygen, that means acidic environment, and high fluid pressure. And all these things fuel abnormal blood vessel formation, inflammation, immunosuppression, fibrosis, resistance to every known therapy, radiation, chemo, immunotherapy, induction of what are known as cancer stem cells. It increases endothelial mesenchymal transition, increases invasion and metastasis, confers resistance to death, changes metabolism, and causes genomic instability and other issues. So you can imagine simple thing like dysfunctional blood vessels of a tumor can create these 8 problems in a solid tumor. Whether they're adult solid tumors or pediatric, they all have this problem. So you can imagine if you were to develop a pharmaceutical company and you were to develop a drug, and if you want to cure, overcome each of these challenges, you'll need 8 drugs. And each of these drugs costs $100,000 a year. You need $800,000. But what should be said to ourselves? So why don't we go and repair the blood vessels, fix the blood vessels of a tumor so that we can alleviate hypoxia, fix pH, and lower pressure? And if you could do this, then simple one drug that can repair the blood vessels can overcome the problems, which will be solved by 8 drugs. And what is also we recognize is that if you have hypoxia and low pH, the tumor microenvironment becomes immunosuppressive. That means the immune cells that come into the tumor stop killing cancer cells. So that's another bad news. So our overarching hypothesis, our concept is the following, which is that if you are to repair the blood vessels of the tumor, improve the function of tumor vessels in a solid tumor in both adults and in pediatric patients, we would increase the tumor perfusion, means blood flow, improve oxygenation, improve the delivery of T cells, these are the cells that kill cancer cells, decrease or increase anti-tumor myeloid cells, and many other advantages, and the microenvironment of the tumor will become immunosupportive as compared to immunosuppressive. So how do we do this? So our laboratory is known for discovering two different strategies to repair the blood vessels of the tumor. One we refer to as vascular normalization. Second one we refer to as extracellular matrix normalization or stroma normalization. So let me show you the basic concept. So to understand these two ideas, these two strategies, think about you're watering your lawn, and this is still very hot in India and US and everywhere in the world, in the Northern Hemisphere. So we all need to water our lawn and plants. You're watering your lawn and suddenly water stops flowing. So what are the potential reasons for it? Well, one reason is that your daughter or your Mali decided to come and bring his or her bicycle and put it on top of this hose. And now the water will stop flowing. This is exactly what happens in cancer and solid tumors. So over the several decades, we have figured out what this bicycle tire is, and more importantly, how to remove it so the blood flow can resume. And what we essentially found out was that the blood vessels in tumors are compressed by cancer cells, by the stromal cells, and by the matrix. So we said, okay, how do we remove the matrix? And I will not go into the details, but over 20 years of work has shown that losartan, which is a generic antihypertensive drug given to more than 20 million people worldwide every year. And we found out that this drug, which is very safe and inexpensive, can indeed accomplish this. I'll not go into the molecular biology, but it can reduce collagen and hyaluronan. These are the two main components of the matrix. And we demonstrated this in a series of studies in animals and then also in patients. So this is, for example, a highly desmoplastic tumor. That means a tumor full of matrix growing in a tumor. And the blue color represents the matrix, and green color represents the functional blood vessels. So notice, wherever you have a lot of blue color, you don't see any green color because the vessels are collapsed, they're compressed, similar to that hose I showed you, the water pipe I showed you. Now, if you give losartan, see the blue color goes down, and now see the blood vessels begin to function here. And what we found out in 2013 in this publication, that angiotensin blocker, that means losartan, can enhance blood supply, improve drug delivery, and improve the outcome of chemotherapy. And subsequently, we have shown this for immunotherapy also. So, and we have taken this idea from bench to bedside, and I'll not discuss that in the interest of time, but I'll be happy to discuss that later. But I'll spend a lot of time in normalizing vessels using another concept. And that is, again, let's go back to the hose. And the water is flowing nicely and suddenly stops flowing. And you look on there's nobody putting a bicycle or their foot here. So what are the other possibilities why the water will stop flowing? Well, one possibility is that somebody poked a lot of holes in this hose, made it very leaky. And how do you do that? Well, you take a knife or something, or some sharp object, and begin to poke holes. And that sharp object in our body is a molecule known as vascular endothelial growth factor, VEGF. And that leads to this leakiness, except that tumors have a lot of this protein, VEGF. And as a result, the tumor vessels are leaky throughout here. And what that does is blood flow begins to shut down. Now, this molecule was originally targeted by drugs such as Avastin to cut off the blood supply of the tumor, but that did not work. What worked was when you combine it with anti-VEGF therapy with other drugs such as chemotherapy or radiation therapy or immunotherapy. So what's going on here? Why is this drug working with that? And the idea we came up with was that if you use lower doses of this drug instead of the very high dose, then you remove enough VEGF so the vessel leakiness goes down and flow resumes. So this idea was very, very counterintuitive. It was 180 degrees of the current times paradigm. Because when blood vessels, where people were trying to cut off the blood supply by giving drugs that target VEGF. And we said, no, we need to target it at lower doses so we can resume blood supply. So I had a lot of trouble having my grants funded. I had trouble getting my paper for 5 years. So we said, okay, let's collect the evidence. So we did. And this is a breast cancer growing in a mouse, and a mouse's breast. And what we're seeing are blood vessels every 2 hours after we give an anti-VEGF therapy, an anti-angiogenic therapy. And you see the vessels begin to get straighter, narrower, and what we found out, the blood flow went up. And then when that happened, you could deliver more drugs. And the tumors would respond, and mice would be cured, and they would live longer. So when we finished and published this work, the next criticism was, well, this is all in mice. Does it happen in patients? So to answer this question, I started 40 clinical trials with Harvard doctors, both at Mass General and Dana-Farber Cancer Institute, in rectal cancer, brain cancer patients, and breast cancer, GI, sarcoma, ovarian cancer, schwannoma, and pediatric brain tumor. I'll talk about that later towards the very end of my talk. And we tried various anti-VEGF drugs, which were undergoing clinical trials. And essentially, the message we got was, if you gave the same dose of drug to a cancer patient, in this case, a patient who has brain tumor, you find out that in about 20 patients, the blood flow actually goes up. And about 10, it doesn't change. And about 10, it goes down. So if you had come to this lecture before 2001, and I had asked you the question, which of these patients are going to live longer? You would have said these patients. You would have said, sorry, these patients, because you are starving them. You're lowering the blood supply. But so they cannot grow. But what we found out was that these patients were living 9 months longer because of what I just proposed. That when you do this, you can bring more drug into the tumor, and therefore the patients respond better. And that's why in these patients, the survival will increase. And now we have seen this in a number of tumors, different glioblastoma, recurrent and newly diagnosed, lung cancer, breast cancer, both triple negative breast cancer and other types of breast cancer. So then we asked the question, can we improve immunotherapy by repairing the blood vessels of the tumor? But the word we coined was normalizing tumor vessels or reprogramming tumor vessels. And this question was answered by a postdoctoral fellow of mine who's now a professor in China, and published 12 years ago, where we showed in breast cancer in mice that indeed, if you have receptor 2 antibody, you can increase perfusion, and you can decrease hypoxia, and you can improve the delivery of CD8 cells. These are the cells that kill cancer cells, and you can make macrophages anti-tumor. And when you combine immunotherapy, the number of mice cured doubles. So this was very exciting, as you can imagine, for us. And in the meantime, this has been now confirmed by hundreds of laboratories worldwide. But more importantly, the US FDA has approved 7 combinations of anti-VEGF drugs with immune checkpoint blockers, such as anti-PD-1 and anti-PD-L1, in the last 5-7 years. For lung cancer, for renal cancer, for liver cancer, and for endometrial cancer. And right now, there are hundreds of trials going worldwide. So we can imagine that this is going to benefit more cancer patients, this combinations. So you can imagine for a scientist, this is very gratifying. So, but here comes the very sad news that this approach and these immune checkpoint blockers that have been approved now for 70 different indications in the United States, they do not help glioblastoma, the most deadly brain tumor. And all three randomized trials in recurrent glioblastoma and in newly diagnosed glioblastoma have failed. And even a trial, we did a phase 2 randomized trial in recurrent glioblastoma, where we combined pembrolizumab with bevacizumab, that also failed. So currently, there is no treatment, and as I know you treat pediatric brain tumors, but currently we really not made any difference in adult glioblastoma. And as all of you know, problem remains the same in many pediatric brain tumors, such as group 3 medulloblastoma, where we cannot make any difference whatsoever in the lives of these patients currently. So what I'm going to do is very quickly go over why these therapies fail in glioblastoma patients. I'll go through it quickly because you may not be interested in it so much because these are adult patients, but these principles can be conveyed to pediatric brain tumors. So one reason is that when you give these drugs, such as immune checkpoint blockers, they increase edema. And when you give edema, to control edema, you give steroids, which are immunosuppressive. So you're essentially killing positive with a negative. Second is that brain tumors, whether they're primary brain tumors or metastatic brain tumors or pediatric brain tumors, they have a type of cells in their brain known as the regulatory T cells that are highly immunosuppressive. And finally, brain tumors have a protein called angiopoietin-2, which is also highly immunosuppressive. And it goes up further when you give anti-VEGF therapy. So let me just quickly go over our strategies to overcome these challenges. In the interest of time, I'll cut short this part of the talk because unfortunately I put too many slides. So this is a glioblastoma patient. You can see this edema very clearly in the brain of this patient prior to an immune checkpoint blockade. But once you give this patient immune checkpoint blocker, anti-PD-1 antibody, you see the edema goes up. Now what you have to do is give it a steroid. And when you give steroids, like such as dexamethasone, they're highly immunosuppressive, and the survival goes down of these patients. The reason for that is the entry of these various immune cells goes down. So about 10, 15 years ago, a French physician, Dr. Carpentier, showed that losartan can control edema in GBM patients. But last sentence of each of his studies, the mechanism of edema reduction is not known. So that's when we said, okay, if you want to use this in GBM patients, we have to know the mechanism. We need to know how losartan is helping. So I asked one of my PhD student, Dr. Meenal Datta. She is now an assistant professor at Notre Dame University. And she grew glioblastoma in the brain of mice. And then she gave anti-PD-1 antibody. And you can see these vessels become leaky. And the edema goes up similar to what happens in patients. So when she gave losartan to these mice that had different brain tumors in them, this one versus this one, you saw losartan alone decrease edema. But when you give this anti-PD-1 antibody, edema went up, similar to what we saw in patients. And when she combined losartan with anti-PD-1, edema went down in both tumor types. So the next question is, why is a blood pressure controlling drug is controlling edema? How is it doing that? I'll not go into the highly sophisticated molecular biology approach to do this, but essentially to give you the bottom line, the conclusion that what anti-PD-1 antibody was doing was upregulating, increasing the expression of two different genes in the endothelial cells of the brain tumor, MT1 and MT2, these two genes are involved in disrupting blood-brain barrier. The blood-brain barrier becomes leaky when you have this, and that begins to explain. So when you give losartan, notice that this does not go up. Only with anti-PD-1 it goes up, but when you give it losartan, it goes down. And so that's exactly why we saw the benefit. So then we asked the question, what else does losartan do? How else does it benefit these patients or these mice? So again, what we found out, we went back and looked at all our glioblastoma patient data at Mass General Hospital and found out that cancer patients who were taking antihypertensive drug of this class, they had increased blood volume and increased perfusion, similar to what we had suspected and seen in our animal studies. So then we asked the question, does it actually happen? So here's a glioblastoma growing in the brain of a mouse. These red color represents the functional blood vessels that are perfused with blood. And now if you give losartan, you see that the vessels become straighter, narrower, more close to normal vessels, and the perfusion goes up. And then we asked the question, what else is losartan doing? And what we found out was that it's reducing the matrix, hyaluronic acid, which crosses the blood vessels, and it decreasing the swelling in the tumor. And this is very mechanistic, but essentially it's polarizing the myeloid cells in the brain, so they become anti-tumor. And it's increasing the ratio of killer cells to the regulatory T cells. And this is really the benefit mechanism behind it. And when we combine losartan with, losartan alone doesn't do anything, but when we combine it with, when we give anti-PD-1 antibody, it doesn't, there's no long-term survivor. But when we combine with losartan, you see that 22% of mice are cured with this tumor and 15% mice with this. And now if you combine this with the standard of care, which is surgery followed by radiation and chemotherapy, you see that the number of mice being cured goes up all the way to 43%. And if you challenge these mice with another tumor, they are rejected, suggesting that these mice have developed a memory response against tumors. So next question was, how do we distinguish responders from non-responders? Can we find biomarkers that we can use to select these cancer patients? And indeed, we have been able to do that. The challenge, of course, is that by the time a treatment outcome can be assessed, it's too late to identify what contributed to it. So Dr. Meenal Datta did is she implanted these glioblastoma bilaterally, one on each side of the brain of a mouse. She resected one. So we knew what the baseline was like, and then treated mice and then looked for the signature. And essentially what she found out was the animals that survived, that got cured, had high number of immune cells at the baseline. So this is, we're planning a clinical trial now with losartan where we'll use this as a baseline. So what I conveyed to you, you don't have to understand the molecular biology, but what we essentially showed was that losartan, the reason it benefits immune therapy, is reduces the extracellular matrix and the compressive stress. It decompresses and normalizes blood vessels. Remember the bicycle tire, removes it. It improves the edema, it controls the edema and promotes immune stimulation and reduces immunosuppression and improves survival. And the tumor microenvironment baseline can predict long-term survival. So that's mechanism number one. Second mechanism, abundance of regulatory T cells. As I said to you, these are the cells that have a negative effect on in cancer, and they are the ones that stop the function of killer cells. And if you look at, we have looked at a variety of primary glioblastoma and MYC-driven medulloblastoma, metastases from a triple-negative breast cancer, metastases from HER2-positive cancer. In all cases, there's abundance of these highly immunosuppressive molecules in the tumor. But what is really worse is then when we treat these mice with an anti-PD-1 antibody, which is supposed to improve immune response, actually these cells become even more immunosuppressive. So can you imagine this? You're giving a drug to improve the immune response. It's doing the opposite because of these cells. So what do we? So again, to summarize, these regulatory T cells contribute to inherent resistance to immunotherapy. And then when you combine with an immunotherapy, they also develop acquired resistance. So you're doing a double whammy. You're doing these cells are really doing two different damages to the treatment. So our thinking was that why don't we reprogram these cells? So convert them from cells which are immunosuppressive to immunostimulatory and see what happens. Does it restore the function? And I will not go into the details of molecular biology to find out what molecule on these cells to target. We used a technology known as single cell sequencing. And we found out that one specific molecule known as GITR is highly expressed in the glioblastoma regulatory cells compared to systemic regulatory T cells. And then we found out that when we combine this with an antibody that activates this molecule with the immune checkpoint blockers, we can reprogram the immune cells into killer cells. And we can improve the survival immensely, as shown in this slide. By 20% mice are cured. And then when you combine the standard of care, 40% mice are cured. And if you re-challenge these mice with another tumor, it's rejected. And what was nice about this, that the memory response was confined to just near the lymph nodes, cervical lymph nodes. So that means there were no systemic toxicity. So conclusion of this part is that these cells, which are abundant in glioblastoma, are highly immunosuppressive, and immune therapy can make them even more immunosuppressive. What we showed was that activating a molecule which is upregulated in these cells can reprogram them to kill more cells. And this improves immunotherapy and cures mice. And finally, as I mentioned to you, that there's a third reason why immunotherapy failed in glioblastoma patients, because they had a protein which is highly immunosuppressive. This is known as angiopoietin-2. And you see in newly diagnosed glioblastoma and in recurrent glioblastoma, this is a log scale. Notice that it's very highly expressed, and this is immunohistochemistry. You can see it, normal brain versus glioblastoma. So what is worse is, and we had shown this in patients, that after we give them anti-VEGF therapy, it goes down for a little bit and then goes back up. So this is where the problem begins. So when we blocked both VEGF and anti-angio2 in mice, we found out that the immune cells began to infiltrate the GBM, glioblastoma. See, the CD8 cells went up compared to control of IDB. And so the next question was, why does this happen? Why do they go in? And the answer is that this therapy, by normalizing the function of blood vessels, improving the function of vessels, increases the perfusion and decreases the hypoxia. And that in turn brings in more immune cells into the tumor. And now there is a type of molecule which immune cells need to get into the tumor that also goes up. So as a result, when we combine this approach, 20% of mice are cured with such an approach. So to summarize this challenge, this particular antibody that blocks both VEGF and angiopoietin-2 converts T cells into antigen-presenting cells, increases adhesion and function of T cells, and enhances their activation and memory, and improves survival. So as we are finishing this work, we ask the question, can improving the function of vessels improve CAR-T therapy? I'm sure you've heard about CAR-T therapy. It's a therapy, especially for hematologic malignancies. It's been approved. It's revolutionized the treatment. And now, and India is now making very inexpensive CAR-T cells for treatment in patients. And Mumbai is a big center for that, for this technology. And as you probably have been following the trials even in India, that it's working there also, although it's very expensive. And that's why I'm very gratified to see the Indian companies and academic centers are working on making this more affordable. So we asked the question, can we make this therapy more effective against glioblastoma, against brain tumors, by repairing the blood vessels? And I'll not go into the details, but what I want to point out that one of the molecules that's upregulated in glioblastoma is EGFR variant 3. And essentially, their CAR T cells are engineered to target this. And notice that they don't get into the tumor. But if you treat these tumors with this low-dose anti-VEGF therapy, you see there's an improvement in the delivery of CAR T cells. And not only that, and these CAR T cells are very active. That means they kill cancer cells more effectively. And the T cells of the host also gets into the tumor, and they become more active. And as a result, when you combine this approach, you see that in a brain tumor, there's a very impressive survival improvement in one brain tumor type, a second brain tumor type. And currently, we are planning to test this in pediatric brain tumors. So essentially, what I conveyed here to you is the abnormal vasculature and immunosuppressive microenvironment limit the CAR T cell delivery, infiltration, and efficacy. And if you use the right dose of anti-VEGF therapy, we improve the CAR T cell infiltration and efficacy by normalizing tumor vessels. So what I've done so far in my talk is discussed multiple barriers to treatment in solid tumors. They include physical barriers like the high interstitial fluid pressure or compression of blood vessels. And what I want to point out to you that this whole concept of vascular normalization, a number of diseases, not only cancer, a lot of other diseases that have normal blood vessels, and that affect about 500 million people worldwide. Let me just give you 3 examples. One is NF2 schwannoma. These are benign tumors that grow in the ear of patients, young patients. And I'm sure you have seen these patients in your practice. And these patients lose their hearing. And surgery and radiation does not improve their hearing. But what we found out was that when we treated these tumors with the low dose of bevacizumab or Avastin, the hearing came back. This work was published in the New England Journal of Medicine in 2009. And you can imagine if you're a child, your hearing has been impaired all your life, and suddenly you begin to hear things. You can imagine the joy it gives you and to the patient and to the physician who's treating you. And as a result of this work, this treatment was approved in United Kingdom, in England, in 2014, and many other countries after that. We also found out that losartan can also do the same. It can restore hearing. This is the work done in mouse in collaboration with my colleague, Dr. Leishu, and published in 2021. And currently, we are doing a clinical trial of this planning to do a clinical trial of this in patients at Mass General Hospital. Finally, I want to give you the example of tuberculosis. You know, India is one of the leading countries where the tuberculosis kills a very large number of people. And as you know, currently the TB drugs do not work. They fail to improve survival in these patients. And more than 1 billion people worldwide have tuberculosis. And the whole focus of tuberculosis research in the world has been on the bacteria. But we asked the question, does the drug get into the granulomas or in the TB lesions? And as shown here, it does not. It remains on the margin, similar to what we saw in the tumor. So you can imagine that's why TB drugs don't work. But what we found out that when we give losartan to these animal models rabbits that have granulomas similar to what we see in patients, we see that the very recently approved tuberculosis drug, bedaquiline, gets him. And he goes up eightfold. And this work was just published recently, and currently so it was supported by the Gates Foundation. And we are currently discussing a clinical trial for losartan in tuberculosis patients, either in South Korea or India, and with the support of the Gates Foundation. So I hope what I have been able to convey to you so far in my lecture is that the blood vessels and tumors are abnormal, solid tumors, and they create many kinds of trouble. I discussed those troubles with you. Physical forces in tumors are also troublemaker, fluid pressure, solid pressure, and they create trouble. And what I've done is shown you two different strategies our laboratory has developed: vascular normalization and matrix normalization, that can convert this abnormal tumor microenvironment to more reprogrammed or normalized tumor microenvironment. And I have shown you how we have taken these findings from bench to bedside, not only in the tumor, but also in non-malignant. I just want to end my presentation by showing you our work on pediatric brain tumors. So I will not, since all of you are pediatrician, I'll not give you the background, but one of the major problem in pediatric brain tumors is glioblastoma, and especially group 3 glioblastoma. And currently, there's no treatment for group 3 glioblastoma. So in 2013, 11 years ago, we showed that if you target a molecule known as PLGF, placenta growth factor, in mice that have pediatric, so this is a control treatment. And you see that in 6 weeks, so that we implanted medulloblastoma in the brain of these mice, and you see and 6 weeks later, the tumor grows and it spreads through by CSF, similar to what happens in patients, in pediatric medulloblastoma patients. But when we treated these mice with an anti-PLGF antibody, see that 6 weeks later, the tumor has shrunk, and you suddenly do not see any spread via CSF. So this was very gratifying. It was published in New England Journal of Medicine, and I think I'll come to this. And there was an editorial about it in the New England Journal of Medicine in 2013. And indeed, we did a clinical trial with this drug in collaboration with Dr. Scholler, who is a pediatric oncologist in Boston, at Harvard, not at Harvard, but in the US. And we did a phase 1b trial in not only medulloblastoma patients, but other pediatric tumor patients who had failed 6 to 7 different treatments. And what we found out was that these patients, for example, one girl is living now more than almost 2 years. So what that tells us is that we need to look at the microenvironment, and that's where we're going to find new treatments, not only for adult, but also for the pediatric brain, pediatric tumors. And unfortunately, this company that was making this antibody decided not to pursue this because they do not make money with pediatric tumor patients. They're not enough in numbers. So right now we are looking for some other treatments for these patients that would also work in pediatric and adult patients, so that the pharmaceutical companies would be interested in it. So let me just end my presentation by mentioning to you, by acknowledging my colleagues. This is the Steele Laboratories, and these are my colleagues, my fellows, who have contributed to the research I described to you. This is the team of my Harvard and Mass General glioblastoma collaborators, basic scientists and clinicians, and who are the leaders in the field. These are the colleagues who have done trials in other tumors. And I especially want to acknowledge Dr. Scott Plotkin, who has led the trials in schwannoma, as well as other pediatric brain tumors with us. And I want to point out to you that for the last 39 years, I've been teaching a 4-day course on tumor microenvironment. And this course is this year it will be offered on October 28th to 31. And one of your colleagues from the Birla Tata Institute has actually attended this course in person before COVID. But after COVID, now we have made this course virtual, and it's offered through Harvard Medical School. And I really, really hope you would consider taking this course. It's a 4-day course. And as I said to you, this, and I'll be happy to send you the website of that. The web link is at the bottom of this. Also, it's in our website of our laboratory. And it's the good news is that since it's live, since it's not live, but since it's offered virtually via Zoom platform, we record it, and the recording is available for 1 month for people who are registered for this course. So I hope you and many of your colleagues will join the course this year. And finally, I want to acknowledge that about 250 graduate students and postdoctoral fellows with whom I've had the great pleasure of supervising and mentoring in the last 45 years of my academic career at not only Harvard, but before that at Carnegie Mellon University, and before that at Columbia University. So without them, we would not have done what we have been able to do. And I know several of you have wished to pursue any postdoctoral fellowship opportunities in my laboratory. This is the QAR, and you can take a photo of this, and you can apply to this. And I know I have many fellows from India who come with their own fellowships, and we welcome them. So thank you very much, and I'll be very happy to answer any questions you have. I don't know if that's allowed or not in this platform, but if it is, would be happy to do that. Thank you very much again for your kind invitation.
Thank you so much, Ragesh. I can see Dr. Banavali is on the line too. Sir, do you want to?
Hi, Shripad. See you.
Okay, you're on mute, sir.
Oh, okay. Okay.
No,
Yeah.
Dr. Banavali, you're on mute.
Yeah, sorry. Missed the early part. I was traveling in the
Don't worry
flight,
about
but
it.
so
I
really
know
enjoyed
flight,
the talk.
I know your flight was delayed, so I totally understand it.
Thank you. So, no, but it's a great this thing, and people talk about tumor all the time. The microenvironment is also that important, and I wanted my colleagues to importance of the microenvironment and how we can modulate it to improve the outcomes. So thank you for a great talk.
Thank you. So please, Dr. Banavali, I would be grateful if you could encourage your colleagues. I think you had already sent one to our course in person, but now they wanted to
Definitely.
travel to you.
I have attended it in the past physically, so it's a
Yeah.
great.
But they can now attend it virtually. That's a nice thing. So
Great.
that's, and it because the travel expenses are very high.
Yeah.
But
Absolutely.
yeah. So I would be happy to send you the link for my course, and if you forward it to the people on this, yeah, I'm sure you know Dr. Michael Taylor, who's a leader in pediatric brain tumors. He is one of the faculty members in my course, so he'll be teaching.
Great. Thank you. We'll do that.
Yes. So if there are any questions, I know there are about 4 minutes left, but if not, enjoy your evening, enjoy your dinner. And I hope I get to meet you someday in person when my visit to India.
I think if anybody has any questions, I mean, you can type in the chat box too. So I have a couple of questions. So first of all, I want to say it's a very different and definitely a very, very different perspective from what we are used to as clinicians and clinical oncologists. And the work you have done, I think it makes a lot of sense too, because at a point where we have plateaued with respect to cytotoxic chemotherapy, we have used it in different permutations, combinations, and I think this is where we are stuck now and looking for more beyond and above, and maybe we will see more in future with all these preclinical studies, because they're very interesting to See, now you talked about a lot about anti-VEGF therapy, low dose, low dose. I just wanted to ask, when you say low dose, what dose are we talking about?
So a standard dose of bevacizumab, that's also approved in India. I know that. The standard dose is what, about 5 milligram per week? Yeah, and or about 10 milligram, every other week, or 15 milligrams a month or so. What I'm suggesting is about half or 1/3 of that.
Okay.
That'll also save money,
Okay.
yeah. And just to give you an idea, in the schwannoma patients, right, the dose we gave was about 1/10.
1/10, oh, that's quite.
And hearing came back. Can you imagine a child who's, hearing comes back for the first time? Surgery doesn't do it. As you know, radiation doesn't do it. I mean, he keeps the schwannoma under control, but that's about it. Yeah.
Also, I mean, you talked a lot about how Losartan helps in reducing trauma and how it helps to normalize the tumor vasculature and improve the chemosensitivity of the tumors. So, because and also you talked upon pediatric brain tumors, and the one stroma-rich tumor is medulloblastoma, where you have the desmoplastic blastoma. So, where there is a lot of stroma. So when you take about medulloblastoma, we all talk about the vasculature of Wnt medulloblastoma, where the chemo, because it's very chemosensitive, we can come down on the
Yeah, it
radiation.
is definitely.
All those.
Yeah.
Yeah. So in this desmoplastic medulloblastoma, which is also a favorable one, are there studies on those using this losartan? Maybe there also we can come down on the radiation further, because CSI in children is a huge controversial thing where all their efforts are to minimize the dose and maybe even to
They
avoid.
have to be, because otherwise what you're doing is you're causing lowering of IQ, social problems, psychological problems. Radiation is not good. And so this is why we are looking for therapies that can reduce the dose of radiation. Actually, anti-PLGF antibody does. Since that antibody is not being made, we have now other candidates that do the same thing.
Do you have
I did not have time to,
on medulloblastoma?
yeah, we do. And the right now being written up.
Okay.
And so it should be submitted for publication within a year. And once that does, I would love to do clinical trial in India with Shripa or whoever, because that's not with bevacizumab. No, that's with another drug which has been approved for pediatric tumors. And I am not allowed to disclose that right now because the work is undergoing, but should be ready for public disclosure less than a year.
Sure.
I would love to work with Indian pediatrician. I'll talk to Shripad about it, or whoever deals with solid tumors at your place. I don't know, in medulloblastoma, and because we can lower the dose tremendously of radiation, tremendously.
Sure. I think we're looking forward to that.
Yeah.
Yes. So there is a question from the audience also. Any experience of low-dose bevacizumab in optic glioma, where the vision is severely affected?
No, we have not any work, but I assume this will help, no doubt about it. Look, for macular degeneration, for diabetic retinopathy, low dose Avastin helps.
Yeah.
That's well known.
Yes, exactly. This was the premise behind to use bevacizumab in those with optic glioma, but there the doses are not as low. So whenever we have used the standard dose, the handful of patients we have used it, it has not made much effect or improvement. So maybe we have to look at the dose as you already pointed out.
Yeah, so look at our schwannoma paper, okay? New England Journal of Medicine, 2009, and subsequent papers.
Sure. Yeah.
Okay.
Manas, do you have any?
No, I think it was really an eye-opening talk from the point of view of how bevacizumab works and the concept of low dose, and it actually how we think that it is going to starve the tumor of blood, and you are actually increasing the angiogenesis.
Yeah.
That was really interesting, and
So you're not increasing angiogenesis. All you're doing is making the vessels
increasing the blood
functional. Yeah.
vessels functional.
Yeah.
Yeah,
We're
you are
not
increasing
making
blood
any more.
supply,
Yeah. Yeah.
and then letting the drugs penetrate that area and work.
Yes.
So
Yes.
that was quite interesting. And all these drugs that we use, how they are being repurposed for this purpose. I think that was all very interesting. So thank you for that. You can share the this program of yours, which students can register and
Sure.
do online.
So
We can
who
circulate
who should I
to
send
our.
that to?
So you can send it to Dr. Banavali, or
Okay.
the group email through which we have been chatting to organize this. And through our PHO chapter, we can circulate it to the entire community. And I'm sure that there will be some takers who would be very, very keen to pursue this
Okay.
further. So thanks a
And
lot
could you
for
also forward that to the adult oncologists?
we can
Alright.
do that. We can do that. I think Badira through the CG, maybe, we can,
Yeah, sure.
send a message to everyone about this thing course.
All right. So I'll
Yeah.
send this to Dr. Banavali, and then he will forward you. I'll make a special. Yeah.
We'll
And look,
circulate it.
it was a real pleasure. Okay. Enjoy evening.
Thank you.
Thank
Thank you very
Thank
much.
you. Thank you very much. Bye-bye.
Good day to you.
Yeah. And good night to you.
Thank you. You all.